Hi everyone. So welcome to week 3. I'd recorded this in our Zoom call last week, but unfortunately the audio didn't capture, so I could hear everybody else's input, but I couldn't hear myself in the video, so I'm just going to re-record it fairly quickly so that you guys have access to this. So this is week 3. In week 3 we're going to be going from the ASV clustering, so making those groups of organisms from the sequences, to determining the taxonomy and measuring diversity. Specifically, I'm going to talk about first, how are we actually going to record our steps if we want to show somebody what we did in a publication or something like that. How would we go about logging that? Then we'll continue with the Deblur, determining the different ASVs there. We'll generate some figures if we have enough time, and do differential abundance testing, and then repeat all of this with data too. That would be the ultimate goal. So how can we keep track of our work is obviously going to be a big one, because if I come back in 2021 and look at this folder here, I might be like, okay, I don't really know where to start. And so in QIIME2 we know that we can actually look at our files and then click on the provenance tab here. And the provenance tab lets us click on different areas of the thing, so we could look at what steps we use for the demux portion, what we use to generate these demultiplex sequences, and all of the details will be kept in this area here. So for example, we see the trim length of the file, so we decided in this case that we would cut the sequences off at 250 base pairs. We have the mean errors that we accepted, the indel probability, indel max, and how many jobs to start. So this is all recorded in the provenance tab. So that's great because it stays with the file. So if you had a collaborator that wanted to see some of your QZV or QZA files, you could simply send them that and they could click on these and determine what parameters were run to generate these files. So that's pretty good. Another way that I like though is to actually record my steps on text. So you could use a text editor like Notepad++. This is great because it actually does textual formatting where it's got the spacing properly, and you can tell it what language you're working with. In this case, it's Unix. Let's mark that. And because it's Unix, it'll have different colorations for certain Unix commands like wget, remove, and make directory, and all these other ones down there. So it really shows you that in a visual way, and that can be helpful. And so here I've got the steps or the pipeline for the microbiome data analysis that I did over the summer, from installing Linux to going and unzipping all the files and doing certain cleanup steps. And so this is a good option. Notepad++, highly recommended. It's a free software and it doesn't really take a lot of space in your computer. The other one would be to actually use PowerPoint, and this is what I did with my students over the summer. I had them record their steps in PowerPoint, take screenshots of the different files. So this is the metadata file that I'm showing here. Then they took screenshots of the commands they used to do the import data function to show us the quality of the different reads. So we can see that the quality here is fairly good up until about 140 where you start to see it drop off. And so that is stored in a PowerPoint file. And they can also show you through PowerPoint, the actual outputs. So the visuals without needing to use view.qiime2.org. But my absolute favorite is going to be to actually use an integrated development environment or an IDE. This is pretty much like RStudio if you've done any coding in R, where you've got the commands that can be executed here, the terminals below that are actually the place where the commands get executed, a place where plots can appear, and so that is kind of like a visual way of interfacing with your data and seeing the outputs. And it also allows these handy functions where you can press run, for example, and run the whole file with just one button press. Now, because QIIME2 is usually run on sort of the Unix command interface line using bash, I highly recommend you download Visual Studio Code. And so Visual Studio Code is fairly similar to RStudio, but in this case, it's actually letting me run bash commands, which is awesome. So if you're using Windows 10 and you're using the Linux subsystem, which I'm doing right here, the Windows subsystem for Linux, WSL, and it's running Ubuntu, then it lets you run bash commands right in the text. And so, for example, I could store this file and run conda activate this command with just control enter when I'm on that line. And so that makes things fairly easy, and I can attach that to any files or data sets that I upload so that people can know what I did. And so you can also see in this command here is that I'm actually commenting, the different steps. So here we're going to begin the QIIME data import, and we're going to set the number of cores that our computer has first with this command, etc. So that is super useful. One more useful thing about it is it actually tells you the files in the folder where you are. So here's some sequence files that I have, different microbiome files for different coral samples from Israel, actually. That was cool. So I'd recommend getting this, and it should work on most platforms, although I'm not exactly sure if you'd need a Windows subsystem for a Macintosh, for example. Okay, so now we will go ahead and continue with the ASV generation. So ASVs, last week I think we were able to start with the raw sequences, demultiplex them, and so these blue ones are steps that we're doing, so processes that we're doing, and the red ones are actual files or data sets that we start with. So we start with the raw sequences, we demultiplex. If you have paired in data, you'll join the pairs. Once you do that, you have your demultiplex sequences here. And I think that is where we stop. So what we'll do now is we'll view the output from last section, looking at the demux.qzb file. So forgive all these windows that I have open, and clearly none of them are what I want. So we'll do File Explorer. I'm going to go to our tutorial file. Here it is. Now I've already run a lot of the codes, and so we're going to see all the files here. So here's my demux.qzb file. I'm going to go ahead and make a new window in Chrome. Let me show you that here, and then we'll go to view.qiime2.org, and I will now drop in the demux file. And so this is awesome because it's showing us all of the details from our previous run. So it tells us how many forward reads there were, what the minimum, median, mean, and maximum were, and also the total. So you can see total 260,000. It gives us a frequency histogram, so the number of sequences per file or per sample in this case. And you can see that most of our samples were between 2,500 and 5,000 number of sequences per sample, but some had quite a bit more. And then we have more data on the side here where it actually tells us the sequence counts per sample ID. So here we have the highest frequency count right there. And then as we go further down, we see that the lowest is 1,800. And so those are really things to keep an eye on, because for you to do future sort of statistical comparisons, you want to make sure that the frequency or the forward sequence counts are the same. Because it would be biased to use one that has 18,000 sequences and one that has a tenth of that, right? Because you might be missing some diversity in this sample, or you might be oversampling a specific taxa in this one. And then we can click on the interactive quality plot and see how the quality of our reads changes over time. So on the y-axis, we have our quality score, where anything above 20 is okay. You don't want to go below 20. From 30 upwards is actually going to be the best. And so we're going to want to find a place to cut our reads at from this file. And so luckily we don't have to do any trimming in the beginning because our reads are very high quality up here. So I can actually put my mouse on this, it's highlighted it, and then I can go here and it says position 1 and it tells us all the details about the scores here. So you can see that the second percentile, the lowest sort of score, is 25, and that's actually fairly good. And our median is 40, so high quality reads from this sample set. But as we go further on to about, let's say, 123 in this case, we see that our median is 25, and below that we've got a lot of quality scores that are 2 or less. And so we don't really want that. And so what you'll want to do is go through these and find the quality score that is still acceptable. And so for this one, we're going to choose 120. So 120, this is 121. So 120 has a median that is 28, so that's pretty good. And then above that, of course, the score gets better, and below that, it's worse. Yeah, but 120 seems to be good. If you do want to cut it off a little bit shorter, you can do that as well. But as you can see, from the 25th percentile down, it doesn't get any better, right? You'd have to be cutting your sequences fairly short to keep them all very high quality. And I think the data sets that, or the ASV generating algorithms are going to be fairly robust to those lower values. So 120 seems good. All right, so that is what that file looks like. And we've visualized our output. So the next step is going to be to do quality control. And what we're going to do is more or less remove bad samples and bad reads, certain ones that don't have high enough quality for what we want. And before we do that, we'll want to start QIIME2. So to start QIIME2, you're going to run that command conda activate QIIME2-2020.8. This number, this version might change if you update QIIME, so be aware of that. Then we'll want to activate tab completion for everybody that's running these commands. It's a really handy way to actually make the execution go quicker. So in bash, you're just going to put the command source space tab dash QIIME. And then what you can do in the terminal, once this command is executed, just type in QIIME space I. And after the I, don't do anything. And just press tab. So you'll write QIIME space I and then press tab, and it should autocomplete that and spell out info. And if it does that, then you've successfully activated tab completion, which will help a lot in the future when you've got to like type in very long file names. You'll just press tab and it'll do that for you. So now we'll go ahead and do quality control using Deblur. So you'll kind of activate. Then we'll change to the moving pictures tutorial directory, and then we'll run this command here, QIIME quality filter Q score with all these different values, and you can follow along. Pretty much all we're doing right now is going through the moving pictures tutorial on QIIME2 doc. So tutorials, and we are doing the moving pictures. So you can follow along here. We've obtained the data already. We've demultiplexed the sequences. This is option 1 for data 2 that you're welcome to try, but that's not what we're doing now. Now we're going to do Deblur. Feel free to read these papers to learn more about them. And then we're going to do the quality filter Q score command. And the parameters used in this command are actually determined by this paper from Bokulich et al. In 2013. And if you're not running any standards with your samples, so if you don't have like a microbial standard that you extract DNA from and process the same as all your other samples, you don't need to mess with the code or the parameters that the Q-score filter uses. You can just continue using the defaults that they have. But you might be wondering, like, what are those default settings and how do I figure out what they are? And so to do that, you're just going to go and type in QIIME quality-filter Q-score space -- help. And just pardon me 1 minute while I open up my Visual Studio Code and show you how that is done, or at least what the outputs are and how you might go about interpreting them. So this is the Visual Studio Code. As you can see, I've got all my code written out here. I am currently in the wrong directory, so I'm going to want to open my recent place, QIIME2 tutorial. So it's going to be taking me there now. And here I have my tutorial for the bash code, but that is not the one I want. I want no, might be in the wrong directory. I believe this is the one that I want to do. Nope. I suppose maybe it's this one. Yep, okay, this is it. So here I have all my code, and we were doing the quality control. So this is the command that we're going to run. I'm going to go ahead and copy that, paste it in here. You can do that with a right click typically, and then I'm going to put -- help, and we're going to look at the output. Oh, sorry, gotta actually activate conda first. So while that activates, it's activating right now. This command is essentially going to help us for anything. So if you make a mistake on any of your commands when you're running these files, it's going to tell you that you made a mistake, and it's going to spit out sort of these red arrow codes, and it's going to say, hey, you're missing this command, or you're missing that. And above that, it's actually going to tell you what the command that you wanted to execute does and what the defaults are. And so now that we're in QIIME, we're going to go ahead and press the up key twice and get to our -- help. Once that is, you can see it's running right now. Taking a little bit, we have our output. So -- help will work for any QIIME command at the end. So this is saying that this file takes in an input, a demux, and it's an artifact. So that means it's formatted in the sort of QZA format that QIIME2 works with. And it can be sample data with sequences with quality. It could be paired end sequences with quality, or join sequences with quality. Those are all options that you can have. Sequences with quality means that there's just the forward read. Paired end sequences with quality means that you have the forward and the reverse reads together, but in different files. And then sample data join sequences with quality, that's if you've already joined the paired end data together, and so it's just going to expect one read per sample, if that makes sense. So you're not going to have the forward and reverse strand. You've already joined them. And this is something that I've done, for example, when I've done the paired end joining using a separate program called pair. And then you'll see that this is required, so you have to supply it with that input. But then here we have all the parameters like the pmin quality integer, and the integer is just saying what it is, what it expects. In this case, it's expecting an integer number. And you can see that the default is 4. Below that we have a window, default is 3. The minimum length fraction, so this is like the minimum length of the read that would be accepted if you trimmed it. So in this case, it's saying that if you trim your read below 75% of its original length, it will get rid of that read. It'll throw it out. But you can learn more about what these do by reading the Bocculage paper, and essentially all of these parameters come from that paper. Then you need to tell it the outputs, so you got to say output filtered sequences, output filter stats. These are both required, and that's more or less what that command requires. And with that done, we can now move on to the next bit, which is to actually do the denoise and clustering using Deblur. So we're going to create these ASVs using this command, QIIME. Hey QIIME, we want you to deblur, use denoise 16s. So it's assuming you're working with microbial samples, the V4 region typically. And then we're going to give it the input, which is the demultiplex sequences with demuxfilter.qz as the name of those. We're going to tell it the trim length. In our case, we're choosing 120 based on our quality plot from before. And then if you're smart, or if you looked at the help file, you'll see that there's an option for this code of --p jobs to start and then a number. And what this is allowing you to do is utilize all of the cores on your CPU. And so what you'll want to do is figure out how many cores your CPU has. In my case, I have 12 cores, so I can put a number 12 in there. You might have 8, 4, or 2, depending on what system you're running on. So feel free to put those on there, and that can help speed it up by a lot. So for example, when I was running this code with just one core, it took me about 4.5 minutes. Using all 12 cores, it took 1.5 minutes. So that's pretty significant savings of time there. Tell it the output of representative sequences, deblur.qca, output the table of data, so all the different reads that they've joined together into ASVs and how many there are, output the sample stats, and then output the deblur.stats QCA. And once you do that, you end up creating a feature table and representative sequences. And so now what we'll want to do is just focus on the feature table and visualize and rename the Deblur stats. So for this, we just run the command chime metadata tabulate. We supply it with the metadata input file, which in this case is the demuxfilterstats.qca file. So that has our metadata in it, actually. Then we're going to tell it to output the visualization, give it this name. .qzv means that it's a visualization. QZAs cannot be visualized in a useful way. It's just going to spit out a citation for you. You'll notice that if you ever try to drop in a QZA file. And then we'll do a similar command. In this case, we're going to be looking at the visualizations of the Deblur stats, and I'll show you what those are in a little bit. And then finally, we're going to use this command move. And essentially, what move is doing is we're going to rename the file names. Sorry. So we're going to say move representative sequences deblur.qca, and by not having a path or anything here and just saying rep-seeks.qca, we're saying don't move it anywhere, put it in the same exact directory that you found it from, but give it a new name. And so it does that for both of them. And so why don't we go ahead and look at those visualizations. So demuxfilterstats and Deblur stats. So here's our demuxfilterstats. We're gonna go to moving pictures here. Chime 2. We're gonna make got some internet issues. Alright, there we go. So we're going to go ahead and move that over. Now we can look at the filter stats. So looking at the sample, the total input reads was 11,340. After doing all of the different operations, removing reads that are too short or reads that exceed maximum ambiguous basis, we end up with 9,232 that were high enough quality for Deblur to work on. And so that's more or less what this file is showing you, and you can download this data if you want to. I'm not sure if this will work. It should though. Show in folder. So this is, I like Notepad because you just press this button, edit with Notepad. So sure enough, yep, this is the tab separated values file. You've got the name and all of the values that were reported here. So that's great. Now we're going to look at the second one, which is the Deblur stats.qzv. So close this guy. Deblur stats.qzv. And for those of you that don't know, you can press Alt and Tab to switch programs like that. And here we have the per sample Deblur stats. And you can see that you can click on a column header to sort the table, or you can mouse over a column header to get a description. So if I mouse over this one, it tells me that the fraction of reads which appear to be artifactual, including those below the minimum size threshold. This is computed as reads that hit the artifact plus reads raw minus reads dereplicated divided by reads raw. And for this, you can see that reads raw were 1,343. And then after doing all of the dereplication, Deblur ends up keeping, I believe, 602. Spend some time and look at this yourself, but 602 and 1,343, it's about 50%, so we're nearly there. And so more or less that's what this is saying. But yeah, feel free to mouse over these yourself and make sure that you're confident about what that means. But what is awesome about these tables is that if I actually look at this equation, so I can, for example, just do the calculation real quick. We got 50, our reads raw is 1,343. Actually, I'll do this in Excel. So we need reads hit artifact, reads hit artifact, and in this case, the reads that hit the artifact is 0, so 0. And then we need reads raw, and our reads raw is 1343, 1343 reads raw. And once we have the reads raw, we need to subtract the reads dereplicated. So reads derep right here, 648, 648, reads derep. And then finally, all of that needs to be divided by reads raw. So that means this, right, 0 plus 1343 minus 648, and then all of that divided by the reads raw. So we're going to actually put parentheses there too. And sure enough, 51.517498. So exactly the same as we get there. And so that's saying the fraction artifact with minimum size. All right, so that means about half or more are gotten rid of. And so these are good things to know. And with that, feel free to explore this table some more on your own. But that is what this visual artifact is. All right, so continuing. Now we're going to create a feature table and feature summary table. So running these commands here. Input is the table, output is the table visualization, and then the metadata file we're going to actually get supply is the sample metadata.tsv, and then we'll have it tabulate a feature table by running this command. And we can give a quick look at those as well. So we're looking at table.qzv. So here's our table.qzv. We'll look at the interactive sequence called sample detail. And what this is showing you is the number of counts. So the feature, how many features were there and how many times were they counted. You can see that the lowest is 514 features and the highest is 6,770. And you'll want to explore this on your own time, but essentially what you want to do is compare the number of samples on the y-axis by the x-axis being the different metadata treatments that you have. So for in this case, we've got our gut, left palm, right palm, and tongue. And then we're going to want to change our sampling depth just to see how that affects the sample size. And you can see that already at 1,000, we've taken the number of right palm samples that are going to be used for data analysis later from 9 to 3. And the more we do this, the more that affects certain files. So for example, if we wanted to include the left palm in our analysis, we shouldn't go above 2,500 about. If we go above that, now all of a sudden we only have 2 samples. And you know that might not be enough for the analysis that you want to do. In fact, in some cases, right, you might want to have replicates of 5. And in this case, that means your minimum sampling depth would about be 766. at least for comparing body sites. For this tutorial, we end up actually going to 1,000. And yeah, more or less the idea is that you're going to want to interactively look at this, choose the sampling depth that's kind of a bias. You'll want to have a sampling depth that is as high as possible without affecting your statistical analyses later down the road. A good way to sort of figure out what the appropriate sampling depth is later is to do an alpha diversity rarefaction curve. And we're going to be doing that towards the end of our tutorial. But for now, the table.qzv file shows you the sampling depth. You can change it and see which samples are removed. So in this case, these samples would be removed from the sampling depth I've just chosen here. So that is the table.qzv file. Now we will look at the representative sequences file. So repseqs.qzv. And you can see this is, I mean, again, it's pretty awesome. You have the different statistics on the different sequence lengths. So you can see that there's 485 sequence counts of sequences, and they all have the same length. And this is because for Deblur, we have to have all of our sequences at the same length. That's the only way Deblur works. Now, data 2, on the other hand, can actually be more variable in the sequence lengths, and so you might actually see different data here. But for Deblur, that's not the case in all of your sequences. So these are more or less our ASVs and the representative sequences that represent that they come from. Yeah, and so if you wanted to figure out what kind of bacteria this is, the people that put together QIIME have this awesome feature. You just click on this link here, and it's going to blast the file for you using NCBI nucleotide blast. It'll take a couple seconds, but not much longer than that. And ta-da, we've got our output. And what it's saying here is that, well, the main thing you want to look at is the name. So it's Bacteroides vulgatus strain, this one right here. And then you'll look at the max score is 270, total score query cover. This is what we care about. It's saying 100% of the query, so all of the nucleotides here matched ours. The percent identity says that, so it's saying the percent identity is 100%, which means that our read there is very likely to be the Bacteroides vulgatus strain. We can blast another one, and it might give us a little bit of a different output. So we'll view the report. Not exactly sure. We might get another sort of 100% match because a lot of like human molecular or human bacterial data sets are fairly well developed. Okay, this is taking a bit. And because they're fairly well developed, that means that there's a fairly nice data set on them. Huh. Just make sure it's not my internet. Oh. Okay, well that's taken quite a while. I'm going to come back to that later if we need to. And so later on, we're going to actually try to take these nucleotide sequences and match them to a database that will download to our computer to determine what bacteria each of these features likely came from. Okay, so we got that. And just so that you guys all know, you actually don't need taxonomy for doing statistics or plotting. So most of it can be done with just those ASVs themselves. So you can get differential abundance, bar plots, and heat maps. You can do diversity analyses and also plot and do statistics on these outputs. If you wanted to do a non-phylogenetic sort of analysis without matching it to any data set, you would run this command, QIIME diversity core metrics. Simple. Input the table, say the sampling depth, which was about 1,102, I want to say. Supply the metadata file, and then just in this case say the output directory. So the output directory, this command is going to create a folder wherever you're working from, and in this case it's going to call the folder core-metrics-results-non-philo, and in that folder is where all the sort of statistical comparisons are going to be placed. And to determine what the sampling depth is, this is just a reminder, you can look at the table.qzb file to determine the sampling depth. Now, what's really great about QIIME2 is it makes the phylogenetic diversity analysis fairly simple. And what that means is that we're going to be taking into account the different distances that each sequence has. So each ASV will have between them. And those differences can indicate something about the function that the bacteria might have, and can tell us a little bit more about, how diverse our samples are, right? Because it's not really that diverse if you have one genus and 10 species. That might not be that diverse because they're all from the same genus. But it could be more diverse if you have, for example, 3 genera and 3 species each. That would be something that might be more diverse. I hope that makes sense. And so the phylogenetic diversity analysis will let you do that. And QIIME2 is going to teach us how to do the faith metric. So with that, we'll work with our representative sequences table. We're going to do some alignment in there. We're going to take those aligned sequences and from them determine the phylogeny, and that phylogeny will then be used to do diversity analysis. So QIIME phylogeny assignment, it's going to say, hey QIIME, do phylogeny. We're going to do this command, align to tree, mapped fast tree. So it's using different programs in here, one of them being fast tree. You input the sequences, output the alignments, output the masked alignments, output an unrooted tree and a rooted tree. And as you can see, these are all QZA files, which means that they're going to be used by different programs later to generate some visual outputs. Once you have done that, so there's not going to be any visual output from this code. So once we've done that, we can actually do a phylogeny with diversity included. And the one that we're going to be using is Faith's phylogenetic diversity metric. And this is actually the one that's going to be weighing the phylogenetic distance. And so that's if you have, there's 3 genera in your sample versus 10 species from the same genus. Well, those 3 genera are going to be more diverse in Faith's metric than in something like a Shannon-Wheeler, which is just looking at how many species there are. So to do that, you'll just run this command here. The sampling depth for this one is 1,103. And it's going to say output directory, core metrics, results. And that's pretty much it. So you can see that we're using that rooted tree from the previous step. So let's look at the output from that. So we're going to go ahead and look at the core metrics results folder. Here it is on my computer. And you can see it is just full of different files. We've got QZA files, QZB. I'm just going to go ahead and sort by Qs by file type. So QZV, these are all the ones we can visualize. We've got a Brake-Curtis distance measurement. So these are mostly anything with emperor as a principal coordinate analysis plot. And in this case, we're using the Brake-Curtis distance. Then here's a measure of evenness. Here's face PD significance. So we're going to look at those in just a little bit. Then we'll do some alpha diversity testing. Alpha diversity is just typically how many species there are, but in this case we're going to use the face diversity metric and actually do a statistical test to see if there's a significant difference in face diversity by sample. And it's going to output that as the face PD group significance. And then we're going to do one for evenness. I'm not exactly sure if evenness takes into account those distances. Generally what evenness is, let's say that you have a sample and there's four different taxa or 4 different ASVs that you find. And each of those ASVs has a very similar abundance or frequency. So let's say they all have a frequency of 1,000. Now that would represent a sample that is very even, right? Because it's kind of like they're sharing that pie. Each has a very similar size slice. But in some organisms like corals, for example, they'll have one dominant group, which might mean that, this group is a symbiotic organism that the coral relies heavily on, or it could be that the coral is being infected by a certain bacteria. And in that case, let's say for this coral, you would have 4 ASVs as well, but one of them has, 3,000 reads, while the others, or at least a frequency of 3,000, so 3,000, while the others are more like 100 each. Now that would be a very uneven community, and for the evenness value, it would be closer to 0 than it would be 1. So 1 means that, everybody's sharing the pie sort of equally, and a 0 would mean that it's really biased to one group. Kind of like the distribution of wealth in America. No big deal, right? So there we go, evenness group significance. And why don't we look at the faiths PD group significance. So there we go. And I'm sorry I'm kind of rushing through this, but I figure this is better than a video that makes no sound, where you can't hear anything. So here we have our faith data, the alpha diversity box plots. If you don't know what a box plot is, go ahead and learn about it. On the y-axis, we've got our faith PD. The higher the value, the more diverse the sample is. Then on the x-axis, we have the different body sites. And so as you can see, the left palm and the right palm seem to have a higher faith diversity than the gut and the tongue. So that's pretty interesting, since the tongue and the gut are kind of connected, right? We might expect that whatever you're swallowing is getting into your gut, or at least it has a similar diversity. We actually don't know what organisms are in the tongue and gut, right? So we can't really compare. Yeah, but are these differences significantly? Now, by looking at a box plot, you can kind of tell. So I could tell, for example, that this gut and the tongue are probably not significantly different from each other, but they're likely to be significantly different from the left palm and the right palm. Whereas the left palm and the right palm probably won't be significantly different from each other, but different from the gut and the tongue. And so a Kruskal-Wallis test is used here, looking at all the groups, and you can see the p-value is significant, so less than 0.05. And then we can actually do a pairwise comparison. So gut versus the left palm, we see, okay, that is a significant difference. So gut versus the left palm, significantly different based on this test. Gut versus right palm, also significantly different. Gut versus tongue, not significantly different, right? That's what we would have expected. And then that's repeated for the left palm. So left palm and right palm are not different from each other, but it's different from the tongue. And then the right palm is different from the tongue as well. And so yeah, we've got some significant differences here. The gut and the tongue seem to have a lower diversity, at least based on Faith's metric, than the left palm and the right palm. And I just want to confirm that I don't have an example here to show you alpha diversity using a Shannon-Wheeler test, for example. So Shannon-Wheeler would be, Shannon-Wiener. Wiener? Yeah. Anyway, a Shannon diversity is not going to take into account the distances as much, so you might get a little bit of a different picture here. But in any case, it doesn't look like I have that file, so we will just breeze on away from there. And we'll look at the evenness for the group significance next. So I'll go make a new page here. Go ahead and get the evenness group significance. Looking at body site again, you can, I forgot to do this, you can look at subjects or reported antibiotic use. That's also an option. And this will all depend on what your metadata is. So from this, the evenness, we can see that we've got low evenness here, higher evenness here, which would mean that, the samples, maybe it's more diverse and they're all about the similar frequency of reads, or something like that. So the higher up you are, the more even, the lower you are, the less even. And just by looking at this by eye, I would say that this probably is not significantly different. And we're just going to go ahead and see if that's true with the Kruskal-Wallis test. So here's our Kruskal-Wallis looking at all groups, and you can already see, this would mean that the test itself is really close to being something that people would accept. But at 0.057, that's usually a value you would reject. And you might want to redo this test with a higher sample size to see if it holds true. But let's say if it did work, then you could go here and compare, and you might say, okay, gut versus right palm, there seems to be a significant difference, and that's pretty much it. So we're fairly close. You want a higher sample size, and maybe it would have helped to have a higher right palm sample, but you know, it's hard to know. So this looks like it's not doing anything, so I'll just close that. So evenness is done. We looked at the outputs. Now we can do beta testing, beta diversity testing. Just one second. Beta diversity is when you're comparing the number of ASVs in one sample to that of another sample. So before we were comparing within an organism, so let's say that those samples came from my body, we were just comparing within my body, and you know, however many times we took samples from it, but we were not comparing it to somebody else's body. Beta diversity is going to be comparing between two different subjects, two different sites, you know, in the world or in the field, or more or less like that. And I recommend you actually go onto YouTube and watch Dan Knight's videos on alpha and beta diversity. He has some really great ones, and I've detailed the links and everything in the syllabus for this workshop. So to run that code, it's pretty much just this: QIIME diversity beta group significance, put the distance matrix in there. We're saying, hey, our distance matrix is in the core metrics results folder directory, right? This slash is saying go inside that folder and then find this file here. And you can see that these are really long. So if you're typing this out, you would start just core dash and then press tab, and you'll probably see it autocomplete this, and it'll go metrics results, and then you'll put this dash, or it might even put the dash for you. Then you just need to type unweighted, press tab, and it might complete this for you as well. So helps reduce the amount of typing that you need to do. Then we're going to give it the metadata. We're going to say, hey, in this metadata, we're going to be comparing body site. So focus on the body site, not subject, for example, or antibiotic use, but body site. And then I'll put this visualization, unweighted unifrac body site significance, and do this in a pairwise manner. And feel free to, you know, put -- help here to learn more about what's happening. And then we're going to do a sort of similar thing. In this case, we're going to be comparing the subject. All right, so body site before, subject this time, and we're going to call that this. So let's go ahead and look at this output. 1, 2. So the first one was unweighted unifrac body site. So unweighted unifrac body site. There we go. So now we've got a larger sample size. Gut's got 28, left palm 56, right palm tongue. You can see, okay, the gut has a lower distance than these ones for the beta diversity comparison. So this is probably saying that these are all not different from each other, but they are different from the gut. And these are the distances, so more or less saying a very sort of similar thing. So the left palm is not different from the right or from the tongue, most likely. And you can confirm all of that later by looking at our Permanova results. So gut, left palm, significantly different. Gut, right palm, significantly different. Gut, tongue, different. Left palm, not different from the right palm, but it is different from the tongue. The right palm is different from the tongue. And so that is what our results are saying. So go ahead, learn more about beta diversity and what these distance values mean. And then the next was going to be comparing subjects. So are the subjects different from each other? Because if they were different from each other, that might be a problem for joining all their data together. And it looks like they are not, and our test should confirm that. So there you go. All right, moving on. Then you can do principal coordinate analysis using the QIIME emperor plot. You give it the data, so here's the PCOA results, and this code is giving us a custom axis, and that's part of the moving pictures tutorial because the moving pictures is supposed to show, subjects take antibiotic pills and then they track those subjects over time, and what you find is that as they progress over time, their microbiome shifts as well, and you can kind of see that in the PCO plot. And so that's what this command is trying to do, and give you custom access to visualize that. And it's doing it twice. So this one for Bray-Curtis, and this one for unweighted unifrac. So feel free to view those on your own if you're curious. I'm not going to do that for this video, but I will go ahead and show you what an alpha rarefaction file is. This is the one that can help you determine how many sequences are needed and what the trade-offs are. And it actually should have been run before we did any statistical comparisons, because this would help you decide, what value to choose. So QIIME diversity alpha rarefaction, input the table, put the phylogeny, the rooted tree. So we didn't use the unrooted tree at all, I don't think. We're going to give it a maximum depth. And I want to kind of draw out what this is doing. So bear with me for just 1 second. Here we go. Go ahead and get a pen. Make it red. So let's say that you've got all your different files, right? We showed this earlier with our histogram. We had like the number of samples and the frequency, where we went up to like 18,000 on this side, and then here we've got like 0, 5,000, 10, etc. And our histogram looked kind of like this, right? We had like a lot in this area, it dropped down, and then there was some here. So the idea is to sort of figure out, okay, if we want to include, 1 second, let me change the color. We want to figure out what the appropriate frequency or sampling depth is, right? If we chose 18,000, that would limit us to just these guys here, which might not be enough sequences, right? If we went a little bit further down, we could have these and these ones, so that's more, but we'd be getting rid of those. And so we need to figure out how many samples are required. And to do that, you can more or less take a random subset of this data set and have the program pick different values. So you could have pick from close to 0, close to 4, 5, and for all of those, plot the alpha, and see how that changes over time. And I'm just going to choose another color here. How that changes as you increase the depth, because it might be like something like, oh, it's kind of variable at the beginning and it increases and flattens out, or it could be like, hey, you only need about 2,000, a sampling depth of 2,000, and after that you've captured all of the alpha diversity. And so with this command, we're essentially, I guess, bootstrapping this, or just kind of doing it on our own, and running the same command for each of these different values and looking at the diversity metrics that come from that, and also how many samples are lost. So for example, if this is the line that ends up working, then that means our sampling depth of right around here is going to be fine, and you're not going to be losing any extra information that this 18,000 sampling depth file might have, right? And so that's what that's going to show us. So we'll look at that now, the alpha rarefaction .qzv. So just open up a new one, and you're going to see it's going to be shockingly identical to my chicken scratch drawing. Alpha rarefaction. It's a QZV file. Okay, so for the alpha rarefaction, I realized that I actually did not run this on my computer. So I'm gonna look at it right here. We'll just view that on my computer. And so here we've got the metrics that you can choose. You can choose the Shannon, observed features, or the faith diversity. So let's just do Shannon for now. And we'll just leave barcode sequence. So these are our different individuals, more or less. The barcode being the barcode used to demultiplex them. And you can see that there's different sequencing depths. So this file, for example, stops at about, what is that, 800? So that would be something that matters here. But you can see for most of them, as you increase the sequencing depth, you're not really getting a higher Shannon value. It becomes flat. And so that's a good sign. That means that probably if you pick this value right here, you'd be fine for the Shannon diversity metric. And for observed features, though, you start to see a little bit of a different picture where as you increase, at least for this one sample, the number of observed features is increasing. But you'll want to make the call here at some point. So maybe this value here. But you're going to have some trade-offs. Right now, it's only going to tell us the number of samples being 1 because we're showing individually. But I'm going to change that in just a little bit. And yeah, sorry about the noise. My housemate just got back. So on the y-axis here, we've got the faith PD, and it's showing a fairly similar picture. You know, the value is increasing for this one red sample for the sequencing depth, but you know, maybe we don't want to look at all the individuals. And let's just look at body site, for example. So face PD is showing us that, okay, a good body site might be right about here. That could be fine, right? Because it's gonna, the gut is showing a fairly good value. The tongue is showing a fairly good value. Do see some change as you go up to above 2000 here. And then I think this is the right palm is a little variable, but you know, it seems like once you get to 2000 or 1000, you can pretty much get the picture of how these are different. And then on the figure or the graph below, what you're seeing is that as you change the sequencing depth, you actually change the number of samples that are in your group. So now for the orange one, which is the right palm, we see that as we go above 1000, we drop to 3 samples being included in that group. And a similar picture shown for the yellow one, which is the tongue, as you go above 2500. And for the most part, the gut is fairly robust as you increase in terms of sample size. So yeah, you could look at this alpha rarefaction plot and get that information from it. And you can compare each of these different sequencing depths if you want to, just by changing that number from, I think it was 1102 to whatever sequencing depth you were interested in. Yeah, so that is what we covered in week 3, and I hope that was helpful for you. Feel free to explore and read more about, read more in depth about what all of these things are, and I will see you in week 4 where we will learn about, or where we'll do the taxonomic analysis and actually figure out what bacteria we have in our samples. All right, well, thank you so much and have a great day. Bye.